Well, thank you so much for inviting me to give this talk about our work in genomic profiling in pediatric neuro-oncology. It's truly a pleasure to be here today with so many friends and colleagues in the audience. Now, I personally think that pediatric brain tumor genetics is fascinating, but I understand it might not be everyone's cup of tea. So I hope this talk will still be informative in that it will describe our approach to lab test deployment and the ongoing process of evaluating clinical utility of tests for the benefit of all of our patients. I have no significant financial interest to disclose. Objectives of this talk. Hopefully by the end of this lecture, you will be able to list some of the major advances in our understanding of the molecular basis of pediatric brain tumors, identify the rationale and potential for next-generation sequencing in pediatric neuro-oncology, and describe how genetic testing can be incorporated into the diagnosis and treatment of our patients. As I said at the beginning, I hope that through this talk you'll gain a better understanding of our approach to test development, test deployment, and ongoing test utilization using pediatric brain tumor genomics as an example. I'll describe two of our research studies, and you may find they sound familiar as many people in this audience have been key collaborators. This is a great article from the New England Journal of Medicine that Dr. Lockwood introduced to me. It discusses the rise of precision medicine, including next-generation sequencing of tumor tissues, as well as the problems with such testing, and also the promise it provides for some patients. Hopefully by the end of this talk, I'll have demonstrated how next-generation sequencing of pediatric brain tumors fits into this framework. So let's start with somatic sequencing. In the past decade, there has really been an explosion in our knowledge of the underlying biology of pediatric brain tumors. There have been dozens of large-scale papers written describing the genetic alterations associated with various cancer subtypes. And with this new knowledge has come a lot of excitement, but also a fair amount of hype. Some people stated that with this new knowledge, we were on the brink of curing all pediatric brain cancers. But as most of us here know, translating basic science studies into clinical reality is more complicated than it seems. And this knowledge is still exciting, but we're left with the challenge of trying to use this knowledge to improve patient care. So as a first step, we decided to take a closer look at the genetics of pediatric brain tumors patients at our own institution. And that brings us to the start of our first study. Several years ago, you may remember, Oncoplex was developed by leaders in this room to identify key alterations in cancer-related genes, particularly those thought to be targetable. And though this panel was initially made with adult-type cancers in mind, we wanted to know how it might be used in pediatric malignancies. At the time, we were looking at an earlier version of the Oncoplex panel, which evaluated alterations in 194 genes. And for this study, we were particularly interested in looking at supratentorial brain tumors of our patients at our institution. We selected patients whose families had signed a separate consent allowing for analysis of tumor tissue. We looked at patients resected between 2008 and 2012 and purposefully only included patients who were no longer on therapy since the results have therapeutic implications. And of course, we only evaluated patients who had sufficient tumor that would not be depleted by this testing. So we ran, this left us with 12 tumor samples to run from 9 patients. And this included both original and relapse from 3 patients. We looked at the selected genetic findings and correlated them with histology and immunohistochemistry. So briefly, here are the clinical characteristics of the patients included in this pilot study. We have age, let's see if I can make the mouse work, age, gender, the historic diagnosis, and you can see that it's quite heterogeneous. And unfortunately, since they are all high-grade tumors, you can see that the survival was very poor, with 7 of 9 patients being dead of their disease, though 2 patients were alive and well off of therapy. Of note, one patient was known to have a cancer predisposition syndrome. That's patient 1, who came from a Leaf-Romaine family. And here is a table summarizing the genetic changes we identified in this study. Now, this is a busy slide, and I'll go over a number of these cases separately. But before I do, I just want to point out that the next-generation sequencing identified key genetic alterations in the majority of tumors studied. And this included many changes with implications for targeted therapy or suspected constitutional cancer predisposition syndromes. I want to go over a few of these cases in greater detail. So this is patient 1's tumor. The original H&E is on the top, whereas the relapse sample is on the bottom, and you can see the copy number variation plots on the right. And this highlights the PDGFRA amplification, which was present in the relapse sample only, while the CDK4 amplification, which is another potentially targetable alteration, was identified in both samples. And for those who aren't used to looking at slides, this middle section is the immunohistochemistry, whereas brown is protein expression, whereas the blue is just a nuclear counterstain. So the top is negative and for PDGFRA, and the bottom, the brown, is positive. Sequencing in this case also identified the previously known pathogenic mutation in TP53. There were a lot of other interesting findings in our second patient's tumor. Here are the original and relapse sample H&Es on the left and the copy number variation plots on the right. And you can see that both the original and relapse samples had NMIC amplification, but the relapse sample also had EGFR amplification. And by sequencing, also harbored an EGFR3 mutation, which is the type often seen in many adult glioblastomas. The histology here correlated well with these genetic changes, and so here we see strong NMIC staining in the tumor cells at both time points, whereas the EGFR is staining on the bottom right at relapse only. Patient 2 also had interesting mutations in CHEK2. There were biallelic mutations. One of the mutations appeared to be germline, and CHEK2 mutations are commonly associated with a modest increase in breast and colon cancer risk. They play a key role in DNA damage response. So this was an interesting finding since we would not have expected it in a brain tumor. A couple other interesting findings. We had 2 other tumors that had identical EGFR in-frame insertions in exon 20. They look similar histologically, and these are well-described activating mutations in non-small cell lung cancers and have been described in fibrous hamartomas of infancy, but they hadn't been previously described in any brain tumors, so this was very interesting to us. Our third patient had a tumor that was very different from all others. It had numerous mutations in almost every gene analyzed, but each mutation was only seen in a small percentage of reads. And this is the pattern you can see in tumors driven by mismatch repair. Also found in essentially all cells were 2 inactivating mutations in MSH2, one corresponding to a germline mutation previously described in Lynch syndrome patients. And this patient had no family history of colon cancer, but based on the next generation sequencing, we determined that this patient had Lynch syndrome and that mismatch repair was driving this tumor's growth. And so here on this slide, you can see the MSH2 immunostaining, which is negative in the tumor cells, while the background vessels and glial cells still retain that brown positive staining. So in summary, we found 1 or more potentially actionable genetic changes in the majority of patients, which was very interesting to us. 7 of 9 patients had mutations associated with pathways that at that time were considered potentially targetable. And we had 4 patients suspected or confirmed with germline cancer predisposition syndromes. There were multiple tumors with mutations in EGFR, PDGFRA, and CDK4, and those are pathways where the therapeutic drugs exist and were in they were in trials at the time. And another important point is that in paired samples, we identified potentially actionable mutations that were seen at relapse that were absent in the original tumors. So we published these results in the Journal of Pediatric and Developmental Pathology. I'd like to thank Colin especially for a lot of this work. And then we were kind of left with, where do we go from here? So we have a clinically validated next-generation sequencing panel, and our oncologists were really excited about ordering this in a wide variety of patients. But the questions arose, who should be tested, and when should this testing be done? So should we test all pediatric tumors with this panel, just pediatric brain tumors, the malignant high-grade tumors, just the relapse tumors, supratentorial tumors only in the brain that were the ones seen in this study, or none? What does the data really tell us about what we should do clinically? So that brings us to some of the problematic parts of precision medicine, and we'll discuss some of these problems next. Our study had a number of limitations. First, it had a relatively small sample size, and it's really difficult to come to any conclusions about anything with such a small number of patients. Also, it was a retrospective study, and that comes with all kinds of biases. So one bias is the problems of using a convenience cohort. I try to keep in mind this really nice rhabdomyosarcoma study, where they found that patients with archived tumor tissues available for research had statistically significant, much better survival than those without tumor available. Now, there's no biologic reason why having archived tissue would predict survival, but that's one of the problems with using a convenience cohort instead of studying all patients prospectively in an unselected, unbiased manner. And then we're looking at the big picture of lab testing. So everyone knows that the US spends more on healthcare than any other country, and this is represented graphically on the right as the percentage of GDP, healthcare spend as a percentage of GDP. Some of the largest increases in spending in recent years have been in lab tests and imaging studies. And within lab testing, no area has grown more than genetic testing, which I believe in our lab has grown at approximately 20% a year. Along with increases in the number of genetic tests ordered, we noticed an increase in patient and family complaints about expensive laboratory tests. At our institution, most genetic tests require insurance pre-authorization. So after this first study, some of our oncologists tried to pre-authorize next-generation sequencing on a few of our patients with malignant brain tumors. But we received letters back from insurance processing saying that our testing was denied due to lack of medical necessity or because testing was considered investigational or for research only. So this means that either the family ends up paying the full amount or the lab ends up paying for it out of our operations budget. And if we're being fair, there is some truth to the claims made by insurance companies. Over the years, numerous biomarker discovery studies have generated an array of omic data, but very few have proven to be useful over the years. The studies to prove usefulness need to be large and ideally show how the results change patient care and outcomes going forward. So to do this, the studies should be prospective, unselected, and the larger the better. And this is an area where I think we as pathologists and laboratory medicine professionals need to do a better job of promoting these studies. It can be logistically challenging and expensive to run studies on clinical utility, and for some reason, it isn't seen as being as interesting as biomarker discovery, but I'd argue that in the long run, for patient care, it's most important. So in the remainder of this talk, I want to tell you about our approach to evaluating test utility. Soon after I started working at Seattle Children's, I became more involved in our laboratory stewardship efforts. And we started a national organization to help address these issues, as you all are probably aware. The mission is to improve lab test ordering, test retrieval, interpretation, and reimbursement. And this organization is PLUGS, which stands for Patient-Centered Laboratory Utilization Guidance Services. And UW is a major collaborator and supporter, so I want to thank you for that. We have a laboratory stewardship committee that meets weekly, and the committee includes pathologists, clinical chemists, laboratory and clinical genetic counselors, medical geneticists, specialty lab managers, and business operations managers. We have a weekly working meeting where we review cases, look at test builds, and have reference lab discussions. And then we also discussed focused interventions or projects looking at specific tests, and so that's what I will be talking about next. So what do we do when we have an expensive test that providers want to order that is also clinically available, but the test has unproven clinical utility in all of the patients that our providers want to order the test on? Well, first, we try to review the literature. In most cases, these new tests don't have a lot of literature to back them up, but in some cases, we get lucky and find some literature that'll help guide us. In all the other situations, we then try to partner with our clinicians. We want to have a good relationship with them to be part of the discussion in test ordering. And we work together to fund prospective testing. We also try to get IRB approval to study, test clinical utility, and then publish the results so that others can learn from our successes and our failures. And so that leads us to our second study. So we prospectively studied the use of next-generation sequencing in all primary pediatric brain tumors evaluated at our institution. Testing was paid for out of research funds, so there was no charge to patients or insurance companies. The study period was from November 2015 to 2016, with a year of clinical follow-up. And we held molecular brain tumor boards monthly to discuss the sequencing results, the pathology, the clinical features, and treatment options. Perhaps it was serendipitous, but during this study, there was a large diagnostic shift where the World Health Organization revised the classification of CNS tumors to finally incorporate molecular findings into tumor diagnosis in addition to routine histopathology. So there's still currently no consensus regarding exact best practices. This keeps evolving, but most places use panels of immunohistochemistry, fluorescent in situ hybridization tests to look for gene alterations, amplifications, or rearrangements. Some places, usually in Europe, they're big into the methylation testing. A lot of places do some version of sequencing testing, but next-generation sequencing has started to gain acceptance as a clinical tool to aid diagnosis and guide treatment in pediatric cancers. So in our study, we use the next version of Oncoplex, which detects alterations in 262 cancer-related genes. And of course, it was in the CAP-accredited, clinically validated laboratory setting. And this is a flow chart of the patients included in our study. So we had 88 patients who had tumor eligible for sequencing. 3 were excluded due to lack of sufficient tissues, leaving 85 patients with tumor available. We had 74 primary tumors, and these included 59 newly diagnosed patients, which were a mixture of low-grade glial tumors, medulloblastomas, other high-grade tumors, so a wide variety of tumor types. We sequenced 15 at radiologic relapse, so our patients are constantly getting MRIs, serial MRIs to look for tumor recurrence, and in this case, the MRI identified that a tumor was regrowing, so we went back to the primary and sequenced those tumors. And then we had 11 recurrent tumors, and those are the cases where a second surgery was done after tumor relapse. And again, all of those groups are a mixture of some of both high and low grade tumors, and I'll go through a few of the different categories. Genetic alterations in this study were considered clinically significant if they met one of the 3 following criteria. If they were alterations that either defined or diagnosed a specific molecular subtype of tumor, if they were alterations which identified a proven therapeutic target, if there are alterations which identify possible deleterious germline mutations associated with a cancer predisposition syndrome. And some of these had multiple of these in a single tumor. So in a nutshell, here are our results. So we found clinically relevant genetic alterations in 80% of our cases. These were found to be disease-defining or modifying in 67% of cases, targetable with an available drug in 53% of cases, and potentially germline associated with cancer predisposition in 36% of our cases. I want to go into a couple of the different groups of tumors next. So the first group is our largest group, and that's our group of low-grade gliomas. So this is a figure that just shows the different alterations that we see in these tumors. And you can see that on the y-axis here are the various genes which we found rearrangements or mutations in, and each column is a separate tumor. So for example, this tumor, the first one, had a BRAF point mutation, whereas these in green had rearrangements in BRAF. And then of course we had other alterations, single copy loss in light blue and copy gain in pink. But you can see that BRAF, the majority of our cases had alterations in BRAF. So just so you're aware, pilocytic astrocytomas are the most common brain tumor that we see in kids, and most of them have this BRAF fusion. These tumors are considered benign, so if our surgeons can completely resect them, there is a good prognosis. But sometimes they can't completely resect them because it would be too morbid, such as causing paralysis or blindness. So even though they're low-grade tumors, they still need to be treated with chemotherapy. And this figure is a representative schematic of this tandem duplication, which results in the fusion of the BRAF kinase domain with the N-terminal regulatory domain of KA1549. This mutant protein is constitutively active and drives tumor growth through the MAP kinase pathway. And it's important to know because this alteration can be treated with MEK inhibitors, but paradoxically, BRAF inhibitors actually can cause these tumors to grow. So if you give a patient with one of these BRAF translocations, a BRAF inhibitor, that can actually cause the tumor growth, which is obviously the opposite of what you want to happen. So it's important to know what type of BRAF mutation is actually present in our low-grade gliomas. And so just this is showing a schematic of our low-grade gliomas. As I said, most had alterations in BRAF. However, 21% had FGFR1 or 3 alterations that were considered potentially targetable, and we identified potential germline mutations in a variety of patients, including NF1, NF2, TP53, and ATM. So I'll go through our high-grade glioma group next. And again, this figure just shows the various genes here listed on the y-axis, and each column is an individual tumor. And you can see just overall that high-grade gliomas often contain mutations in multiple genes, unlike that in our low-grade gliomas. For the purposes of this study, H3, F3, and IDH1 mutations were considered disease-defining according to the 2016 WHO. And the PDGFRA, CDK6 KIT, MET amplifications, and inactivation of MSH2 were considered potentially targetable, where we had quite a few potentially germline mutations, particularly in TP53, which was almost all of the patients in this study. I just want to point out this entity because you may not have heard about it before. It's a newer entity that was first described and categorized in 2016 WHO, but it's called diffuse midline glioma, the H3K27M mutant. These tumors have mutations causing a lysine to be replaced by a methionine at the 27th amino acid position in either H3F3A histone, Hist1, H3B, or one of these other histone genes. And it's primarily seen in pediatric patients with an average are seen in 5 to 11-year-olds. But it's kind of resulted in a diagnostic shift in our group because it can involve any midline brain structures. They're 100% fatal. And so they're considered WHO grade 4, but the histology can be deceptively low grade. So it can look high or low grade. And this is really important because when we're doing frozen sections, the surgeon's often asking us, is it high or low grade, so they can determine how aggressive to be with their surgery. And with these tumors, we just can't say. It could be completely benign appearing, but still be fatal. We do have a way to identify these tumors with immunohistochemistry, but that isn't a rapid process. So it's just been one of those areas of, there's a lot of research. Perhaps cell-free DNA will be able to identify these ahead of time, but that's where we are with these very horrible tumors. I also want to talk really quickly about medulloblastoma subgroups. This is a new now medulloblastomas are really split multiple different ways in the 2016 WHO. There are categories that are defined histologically, but mainly they're being defined by molecular diagnosis. So we have a small group that are the Wnt-activated tumors. These patients tend to do very well, and there are clinical trials at reducing therapy for these patients right now, since they have essentially 100% survival with our current treatments. We have the sonic hedgehog group of tumors, and those are kind of split in the TP53 wild type or mutant, and those have various different prognoses depending on whether or not the TP53 is wild type or not. And then the largest group of all are the group 3 or group 4s, which are lumped together in the current WHO as non-Wnt, non-sonic hedgehog, though I think that we'll be looking to split those groups out in future clinical trials. So most places still try to subgroup these tumors by immunohistochemistry. See, there are various stains and different patterns you can see that help make that diagnosis. So Wnt groups have beta-catenin mutations. Sonic hedgehogs should have GAB1 or filament A immunohistochemistry, and the non-Wnt, non-sonic hedgehog tumors have neither, but honestly, it's very, some of these stains are very tricky and subjective and equivocal. So there's definitely the idea that you do need some additional molecular subtyping, even if you can kind of screen by immunohistochemistry. So this is our group of medulloblastomas in this study, grouped by molecular analysis. So again, we have the various genes here, and each of these cases is a medulloblastoma. The Wnt pathway tumor had the classic beta-catenin mutation, and monosomy 6 are sonic hedgehog patients. Some had patch1 and others had SUFU mutations, and they also could be subclassed by TP53. And this is actually also an interesting, being able to sequence would tell you whether or not you could be eligible for a smooth-end inhibitor, which some of our patients were evaluated for, because depending on where in that pathway the mutation is, some of those patients are expected to not respond to that treatment and others will. So this is just another way of showing our tumors. We did have our 1 beta-catenin mutation, monosomy 6, in the Wnt pathway, and 7 of our patients had the patch1 or SUFU mutations, several of which were germline. Other uncommon tumors I'll just kind of briefly go over. These tumors were a heterogeneous group that really didn't fit into any of the other major groups of tumors. We had a variety of embryonal tumors, rhabdoid, meningioma, schwannomas, and basically all that we could say is that a lot of these tumors had biallelic inactivation of tumor suppressor genes. That was the most common alteration, but these are rare tumors, and we don't really have the numbers to say a lot other than that. So I mentioned that we discussed all these patients as we were going forward at our molecular tumor boards. We reviewed the clinical features, the pathology, and the next generation sequencing. Our attendees included representatives from neuro-oncology, pediatric pathology and molecular pathology, neurosurgery, and our genetic counselors, among others. And the tumor board discussions, we tried to assess whether or not the next generation sequencing test results, did they aid tumor diagnosis or subclassification? Did they identify alterations with a proven therapeutic target? Or did test results identify any possible deleterious germline mutations? And we definitely had situations where sequencing helped make the diagnosis. This was very helpful, especially when we had smaller biopsies where we couldn't evaluate the whole tumor histologically. And this was helpful in those that were considered possible pilocytic grade 1 tumors or a higher grade infiltrative glioma. Histone mutations, so the diffuse midline gliomas H3K27M. So at the beginning of this study in 2015 and 2016, we hadn't validated that particular antibody yet, so the sequencing was the only way that we could make the diagnosis during that time period. Sequencing is also very useful in cases with ambiguous immunohistochemistry. As I mentioned, in some of the medulloblastoma cases, there were very ambiguous staining in some of those markers, so it's very helpful to have sequencing be able to confirm things. So when our colleagues in oncology are ordering one of these tests, they're often most interested in identifying genetic drivers that may respond to targeted therapy. And this has been a big shift in recent years. In the past, patients with a particular diagnosis would be treated with a standard cytotoxic regime regardless of the genetic drivers in that tumor. But now different tumors can be treated with the same drug. We even have FDA-approved drugs that are tumor agnostic, such as the PD-1, PD-L1 inhibitors. And so in our study, this is just listing the mutations that are potentially targetable in our patients. So of course we had a number with BRAF point mutations that be treated with BRAF inhibitors. The largest group of patients were those with the pilocytic astrocytomas that could be treated with a MEK inhibitor. We had a few with FGFR alterations and a variety of others, including those with biallelic MSH2 loss or PMS2 mutations. So looking at how were these tests results used. So overall, we had 53% of our patients who had potential targetable mutations in their tumors. And in 2017, when we were analyzing this, targeted therapy had been given to only 6 patients. 3 were on clinical trials. And you can see the different types here. We had a few on BRAF inhibitors, a couple of which were on trials, 1 patient on a smooth-end inhibitor, 1 patient who had mutations in both PDGFR and MET who had inhibitors to both of those on a different trial. And this is probably for a few reasons. So in some cases, the patient, if they didn't have a relapse or had a complete resection, they were considered cured at the time, so they didn't need therapy even if they did have a targetable alteration. The other reason is that in many of these cases, they were still using the first-line therapy, and targeted therapies were used only for second line. So if the patient relapsed, then they would use those targeted therapies, but they'd start with the cytotoxic therapies first. So it's interesting that reanalyzing this a couple of years later at the end of 2019, we actually had 17% of the patients on the study were treated with targeted therapy. So some had tumor growth or first-line therapies didn't work. So we had another who had a PD-1 inhibitor given, another with a BRAF inhibitor, and then a bunch with MEK inhibitors. And that's really been huge in our field. Like, so many patients are now on MEK inhibitors, and it's there are actually some head-to-head trials that will be going on looking at standard therapy versus MEK in therapy up front. So that's really exciting, and it's definitely changed our field. So on to germline cancer predisposition. Studies have shown that many children who get cancer have a cancer predisposition syndrome. In this study in the New England Journal of Medicine, it looked at mutations in genes associated with autosomal dominant cancer predisposition. And CNS tumors are red in this graph. The authors found that 8% of CNS tumor patients had cancer predisposition syndrome related to one of these 21 genes. So the way our study was designed, only tumor was initially tested, and that could be problematic. So if you look at these 2 patients, they were both patients in our study. Our first patient had pleomorphic xanthoastrocytoma, And a TP53 variant was identified at a variant allele fraction of 0.64. This tumor also had a BRAF V600E, as many of these do. And our second patient was a 23-month-old with a choroid plexus carcinoma, also had a TP53 variant detected at a variant allele fraction of 0.96. So if you looked at these two patients and had to guess which one would have germline cancer predisposition related to Li-Fraumeni, you would pick patient 2. About half of our patients with choroid plexus carcinomas actually do have Li-Fraumeni syndrome. But that turns out not to be the case. That patient 2 did not have a germline mutation. So this was our patient who had this pleomorphic xanthoastrocytoma. It was causing seizures. It was in the left temporal lobe. The tumor recurred. It was debulked, and then focal radiation therapy was planned. But sequencing identified these two alterations, the TP53 and the BRAF V600E mutations. And follow-up blood testing clarified that the TP53 mutation was germline. And I remember this because we were scrambling. It was on a Friday night where the patient was set to start radiation therapy Monday, and we were trying to coordinate changes in care. So instead of radiation for this patient, the patient was started on this BRAF inhibitor, and that's huge to avoid radiation in a Li-Fraumeni patient because high dose radiation can cause a secondary cancer in most of these cases. So years later, the patient has had no evidence of recurrent disease. So that's just a great example of how sequencing can change patient care. So looking at our cancer predisposition possible germline variants as a whole, we saw them in 36% of our cases. So 3 of those patients did have previously known cancer predisposition syndromes prior to tumor testing. Those were a couple NF1 patients and 1 NF2 patient. We had a few patients with possible germline mutations in the tumors, but they died before germline testing could be performed, and a lot of those were the diffuse midline glioma patients. And germline testing was confirmed to identify previously unknown cancer predispositions in 6 of our patients. So we had TP53, MSH2, SUFU, ATM, BAP1, and RAD51D mutations identified. And then 9 of our patients where we thought we suspected possible cancer predisposition, those patients were negative. So this patient was actually right after this study, but it's a good example case. So this was one of our patients with clinical NF1. She presented with a right side facial droop. The MRI revealed this pontine mass, and the H3K27M immunohistochemistry you can see on the bottom is negative. But next generation sequencing revealed a hypermutated tumor with biallelic PMS2 mutations. And so this wasn't actually NF1 at all. This was congenital mismatch repair deficiency syndrome. So very different prognosis, but also the patient was able to be treated with a PD-1 inhibitor with this diagnosis, and family members were able to receive appropriate genetic counseling. There was a sibling who also was called clinical NF1, but actually was found to have a similar congenital mismatch repair deficiency syndrome, so a much, much higher cancer overall risk, and it requires different screening. So in conclusions of this study, this is a very large single institution prospective study which evaluated the clinical next generation sequencing in an unselected cohort of children with CNS neoplasms. We found that targeted next generation sequencing was a useful clinical test that can be used in routine patient care of pediatric brain tumor patients. It can confirm or modify the diagnosis. It can molecularly subclassify tumors, and it can identify various therapeutic targets. It can also screen for possible cancer predisposition syndrome. We did find that testing did not change patient care in cases of completely resected low-grade gliomas or posterior fossa ependymomas. So our current approach, how do we put this into practice? We order Oncoplex in the following situations if the results are needed to help make the diagnosis. If our oncologists are planning to use a targeted therapy, or if testing is paired with peripheral blood to help identify a possible cancer predisposition syndrome in the setting of genetic counseling. And this is, we try to routinely do this for our high-grade glioma patients. However, testing is not currently ordered on our completely resected low-grade gliomas or our posterior fossa ependymomas because this testing just doesn't result in any changes in patient management. So if you're interested in more of the details, you can read up about that in JCO Precision Oncology, where we publish this work. And I hope by now I've showed you how next generation sequencing or precision medicine for our patients, it is personalized. We have somatic sequencing. It can be problematic, but it's also quite promising for a subset of our patients. We do have continued work. So we continue to have monthly molecular tumor boards where we review these cases, and we go back and we audit how the test results are used. As everyone in here probably knows, the Oncoplex panel has recently expanded to over 300 genes, and this includes clinically relevant genetic alterations for our patients. So there are RELA fusions that are commonly seen in our supratentorial ependymomas, MYB fusions in our angiocentric gliomas, and Dicer1 mutations in pineoblastomas, and some other rare CNS tumors. And I also hope this has shown you our approach to evaluation of new laboratory tests. So our approach, we try to review the literature, and when there isn't much literature out there, we partner with our clinicians, we work together to fund prospective testing, get IRB approval to study clinical test utility, and work to publish the results so that others can learn from our successes and our problems. So other tests that I'm not as involved in, but I hear are undergoing evaluation in our lab. So there's rapid exome testing in the NICU. Immune cell function testing, I believe there's a panel where they're looking at the prediction in transplant rejection, and RNA sequencing to better classify variants of uncertain significance. So it takes a lot of people to run any of these studies. These are the people who are involved in the multidisciplinary brain tumor team who see our patients in clinic, and you can see quite a few people who are involved in those each day. I also really want to thank our children's brain tumor precision medicine team. Of course, Dr. Lockwood and Dr. Leary have been quite key in this. We want to thank Dr. Vera Paulson and Dr. Suchia for joining us more recently. They've been great at reading some of our cases and also coming over and participating in our molecular tumor boards. And of course, we thank Shannon, our genetic counselor in the laboratory, and Jeff Stevens, who is our research associate who helps us put all this data together. I really want to acknowledge all of the other Seattle Children's faculty, including those listed here, the University of Washington genetics lab, and who've done all the next generation sequencing and analytics. I want to thank our histology lab and our lab genetic counselors, our sendouts team and lab client services teams, and business operations have also done a lot of work on these projects. And of course, I want to thank our plugs group, many of whom are seen in this picture here. And for funding for our JCO paper, have to thank the Seattle Children's Hospital Focus on Kids Laboratory Guild, the Seattle Children's Hospital Funding Hope Award, and the Division of Neurosurgery. And of course, I want to shout out a special thanks for Dr. Astian for supporting these projects. None of this would have happened without him. So with that, I'd like to take questions. Yes, Dr. True.
Bonnie, wonderful talk. Thanks so much. So in evaluating the costliness of it and the benefits, you ever get into looking at the literature of the effectiveness of a drug that is targeted but the literature doesn't argue that it provides that much difference in outcome.
So the question is looking at drugs that basically would be predicted to be targeted and causing tumor to shrink, but they end up not providing the outcome that we would like. And we've looked at that in some of our cases. The MEK inhibitors, I think that the problem is we're just accruing data at this point, and some of the larger trials are where we'll actually see more of this information. So right now with the MEK inhibitors, we do find that most of our patients do very well on those drugs, and however, there have been a few non-responders, and we really don't know why certain mutations in very standard tumors don't respond the way one would predict. But we're looking at that going forward, and we'll definitely try to get more of that data as we go along. Yes, Jeff.
Thanks, Bonnie. That was great. One of the things, since you mentioned stewardship of these things, and also payer alignment with plugs too, one of the things I've heard from some of the folks who represent insurance companies is, well, they'd like to pay for the tests, like the sequence of the genes that make a difference. And so
Right.
you're identifying ones that make a difference, whether or not it identifies a drug or an inherited thing. But there's 300 and some genes in the panel, and then you have just a few that look like recurrent ones. Do you see that it's in the future you might use the data you've collected to narrow your panel to ones that are more parsimonious or more directed, or is there still some value in sequencing a lot of different things? And how would you justify this?
Yeah, I think that's, so the question is looking at larger panels versus doing a smaller panel with the genes that we know are more relevant, and kind of looking at the payer issues with some of those. And so, basically I do think that we could potentially go to certain smaller panels. In fact, that's one of the things we do in a lot of our low-grade gliomas right now is we'll look at just the BRAF single gene testing, but also some other places do have specific smaller panels, and perhaps that might be the way to go in some cases to align better with payers and with their strategies. But it's really, I think we're in the era where we don't quite know. And then we get those cases where the genes that we find that are altered, it is clinically relevant, but it's not what we would necessarily expect, like the congenital mismatch repair deficiency or the patient with the BRAF mutant tumor that also had the TP53 germline mutation. So I think it's a bit hard to tell where you should draw that line in terms of number of genes.